I want to first begin by introducing this concept of metabolic aging. So a person's metabolic age is a comparison between that person's basal metabolic rate against the average basal metabolic rate for persons of that same age. It can be roughly calculated by this equation at the bottom. That's a very rough estimate. There are much more accurate ways to do this. But on average, the number for men comes out to about 1,800, which translates to you having to intake 1,800 calories per day to maintain weight. Anything more, you gain. Anything less, you lose. Women are around 1,400, if I remember correctly. And of course, this is a rough estimate that can be modified by a great number of things, including nutrition, one's age, disease, including neurodegenerative disease, and especially in the setting of a neurodegenerative disease where you have micro-inflammatory events throughout the brain. This local inflammation can affect a number of cellular functions, including autophagy, meaning the cell can't get rid of its garbage efficiently, creating ER stress, so proteins cannot be folded as they should, and misfolded proteins can't be gotten rid of. Mitochondrial dysfunction leading to reactive oxygen species, and oxidative stress is quite common. RNA stress, meaning you can't translate or transcribe proteins as well as you should. And all of these combinations, all these things combined together lead to a metabolic syndrome and to aging-related diseases. Now, our metabolic rate decreases dramatically, from the time we're 5 years old. It starts to level off a little bit in our 30s, but this is natural, and this is exactly how our metabolism should proceed over time. Females are almost always lower than males, and of course we can boost this metabolic rate up or down. Boosting it up a little seems to be good. Boosting it up too much, of course, is detrimental, and of course the other way is equally detrimental, boosting it too far down. So the basal metabolic rate differences in men and women leads to differences in fat as a percentage of body weight. Typically, women have a higher percentage of fat per body weight compared to men. You can see at age 25, the male to female difference. Men start to catch up at 65, but women are still tend to be higher than men. And this body mass index, or in this case, the body waist circumference, is associated with metabolic syndrome and insulin resistance. The red dots there are the HOMA index of a number of individuals according to age, and the blue dots that are overlaid on that are the body, the waist circumference. So you can see there is a strong association there, but by no means is fat distribution the only contributor to metabolic syndrome or insulin resistance. Today I'll use those terms interchangeably. Glucose intolerance, relative hypertension. I say relative because we just moved the bar for hypertension down. Dyslipidemia, age, sex, ethnicity, subclinical inflammatory events, circulating adipokines, intracellular lipid accumulation in liver, and we heard a little bit about non-alcoholic fatty liver disease yesterday, which can occur when you have high circulating levels of lipids and other conditions, and lipid accumulation in skeletal muscle. Now, over the years, there's been a very strong association with insulin resistance and metabolic syndrome to both the onset and trajectory of Alzheimer's disease. And in HIV, there's been a growing association with insulin resistance, metabolic syndrome, and cognitive impairment. There's a number of studies now that have shown metabolic changes associated with HIV infection and related those to neurocognitive function. If your study's not here, please don't be offended. I just threw a smattering of these up to get a general flavor of what's out there. Roles for obesity, metabolic variables, and diabetes have been associated with neurocognitive disorder. Metabolic changes before and after antiretroviral therapy have been noted. There are some metabolic changes associated with HIV infection and some metabolic changes that are associated with antiretroviral therapies. And of course, the combination of antiretrovirals determines heavily the types of metabolic dysfunctions. I'm going to talk a little bit about one of our studies there, cerebral spinal fluid metabolomics, in which we used energy metabolites to try and get a handle on what pushes cognitive impairment and what pushes cognitive improvement. And that was mentioned yesterday. I think it's equally important to look at things that are associated with maintaining good cognition or cognitive improvement as well as cognitive decline. Abdominal obesity, increased inflammation has been associated with inflammation, immune activation, and cognitive impairment in HIV-infected individuals. And then more recently, that immune activation has been associated with innate immune activation and metabolic comorbidities in older HIV-positive adults. So the association of insulin resistance with neurocognitive impairment in HIV began quite some time ago, back in 2006. And you'll notice there's a common thread to these 4 studies here. They were all led by Victor Valcour. So back in the early 2000s, he was the only one who really cared about this. This was a Haughey cohort shown in the graph on the left there. You can see that there is an association with cognitive impairment. The bars going from left to right are no cognitive impairment, mild cognitive impairment, and frank dementia. The black bars are diabetes, so you can see with frank dementia there's a higher incidence of frank diabetes. And then if you look within that group, there's a relationship of body mass to insulin resistance in that population. He then went on, so he's also shown insulin resistance associated with cognitive impairment in that same cohort. He then worked with Weiss and has been working with that cohort for quite some time. So this occurs, also in women. The HOMA index, which is a insulin resistance index, is associated with cognitive impairment. And then his last study, I think, was really interesting. The BMI was associated with cognitive impairment in such a way that too low of a BMI or too high of a BMI, obese or very low, was associated with worse cognitive performance, and sort of an intermediate good BMI was associated with better performance. And the thinking behind that is that somebody with a very low body mass index is more sick, and once you restore better health, then cognitive improvement occurs. This is something that we recently completed. It's been under review for about the last year now. We're into round 5, right? And a few of you are laughing out there because you've all been there, right? And I think it's going to go this time. So this is looking at subclinical measures of insulin resistance in longitudinal samples from the Charter cohort. This was conducted in collaboration with Scott and a number of people at UCSD. What you can see on going from left to right is our circulating insulin levels in somebody who has stably normal cognition, that is from visit 1 to visit 2, interspersed by about a year. They have stably normal cognition. Then we look at a third visit just to ensure the trajectory is correct. And then declining cognition is defined as someone who declines in cognitive abilities from visit 1 to visit 2. And then we look at a third visit to ensure that that trajectory either stays the same or continues to get worse, just to ensure that there's not a blip in cognitive function there. And you can see at baseline, people who later decline have higher insulin levels. They have higher C-peptide levels. If you look at the C-peptide to insulin molar ratio, that's normal, which suggests the problem is with an overproduction of insulin rather than its clearance. Its clearance appears to be normal. Low HDL, high total cholesterol to HDL ratios, LDL to HDL ratios are also elevated. And what's really important about this is none of these would reach the threshold for individuals to be classified as insulin resistant. These are all sub-threshold differences in insulin sensitivity. We then worked with the MAX cohort. So the previous group was clinically, demographically matched by Scott Latender and Tom Marquette. And then we worked with the MAX cohort to clinically and demographically match those patients with the Charter cohort. We found very similar changes, higher insulin, higher C-peptide, low HDL, et cetera. So it very nicely validated in a second cohort, and we think that's important because these two cohorts are quite different in terms of risk factors, race, general composition of the cohorts. So it seems to hold true that some form of mild insulin resistance or different lower insulin sensitivity can be associated with later cognitive decline. Now, insulin, of course, is a master regulator of energy metabolism. It regulates glucose uptake, glycolysis, glycogen synthesis, protein synthesis, and blocks gluconeogenesis, glucogenolysis, lipolysis, ketogenesis, and proteolysis. And in the central nervous system, surprisingly, there's very little known about how insulin regulates energy metabolism. We know that insulin regulates the uptake of glucose into astrocytes. Neurons are glucose uptake insensitive, so insulin doesn't affect their glucose uptake. And then there's been a number of nice studies that are just summarized over on the right here, where insulin has been shown to have potent anti-inflammatory effects, and it has very neurotrophic effects. So it promotes through mTOR synaptic formation and synaptic strengthening. It blocks apoptosis, and it promotes dendritic outgrowth by cytoskeletal rearrangement. This on the far right is a study that was conducted here in New York by David Volsky and Susan Margello in collaboration with the National NeuroAIDS Tissue Consortium. They conducted a whole genome sequencing analysis on brain tissues from multiple brain regions of HIV-infected patients with and without cognitive impairment. And from that study, I pulled some of the, of course there was a lot of gene changes. And from that study, I pulled those that were associated with energy metabolism. You can see there's a lot of changes in cellular energetics in people with HIV infection and cognitive impairment. Changes in genes that regulate ATP, ADP metabolism, genes that regulate glutamine, glutamate, and glutaminase metabolism, mitochondrial function, glycolysis, and then, of course, insulin signaling. On the left part of the figure there is a rather old study, but it shows just above that blue arrow the basal metabolic rate of somebody who's a healthy control. You can see with HIV infection that basal metabolic rate goes up. And then someone with HIV lipoatrophy, you can see it's significantly higher than control. So there's a lot of evidence that in the setting of HIV infection, there are quite dramatic alterations in energy metabolism. And some time ago, we decided to look at living patients with longitudinal studies. And particularly, we looked at cerebral spinal fluid and conducted metabolomics analysis, at this time by high field strength NMR in collaboration with a group at Oxford. Now we do this by mass spec, which is cheaper and faster. NMR works perfectly fine, but it's expensive and takes time. We pulled out a number of energy metabolites from the cerebral spinal fluid. We then took those energy metabolites and we mixed that information, that biological information, together with clinical and demographic information and conducted some machine learning. Everybody, I think, has heard about different forms of machine learning, big data. This isn't really big data. This is more like moderate data. But these types of approaches are becoming very common now in medicine to try and understand very complex diseases. And what this does here, let's see if I can get this to work. Let me see. There we go. This is a prognostic indicator, so we're looking at baseline changes and their relationship, in this case, to cognitive decline. And what this program does is it takes by importance a series of markers and defines a cutoff. So in this case, you have a nadir CD4 below this value, then your current CD4 is important. If your current CD4 is above this value, then your plasma viral load is important, and et cetera. And finally, you get down to either worsening or stably normal. So it comes up with a series of decisions and cutoffs that ultimately identify somebody as stably normal or impaired. It had a predictive accuracy of 86, sensitivity 96, and specificity that was decent, 76. Then we looked at change over time. So now we're looking at longitudinal change in cognitive function, as we were in the first part of this. But we're also looking at change in clinical measures and change in biological measures, and you can see that reduces the number of nodes required to correctly identify people who are worsening or improving, and it increased our specificity to 96%. Now, what we thought was especially interesting about this is the energy metabolites that popped out of this analysis were citrate, acetate in both models. And if you go back to Biochemistry 101, I still have nightmares about this, acetate and citrate feed into Krebs cycle, right? And when a Krebs cycle, or the citric acid cycle, when the citric acid cycle is overwhelmed and there's too much substrate going into that system and it can't keep up with that metabolic rate, then, oh geez, there we go. And you can't keep up with that metabolic rate, then citrate accumulates and it feeds back on this enzyme called phosphofructokinase, which is the rate-limiting step in glycolysis. So this suggests that people who are worsening or have worsening cognitive function have a very high glycolytic rate. And that makes a lot of sense with some of the brain imaging studies that have been done. This is a study by Linda Chang and Thomas Ernst that was finished in 2013. And they're looking at brain activation here in normal individuals on the left and HIV-infected individuals with cognitive impairment on the right. It's an increasingly complex task. So they're tracking 2 balls, tracking 3 balls, tracking 4 balls. When they're tracking 2 balls, you can see the superior, inferior temporal gyri and parietal lobes are involved. Tracking 3 balls, the activation goes way down. They attribute this to a practice effect. This is the same subjects going from 2 to 3 to 4 balls. And then once you get up to tracking 4 balls, now you have frontal regions and occipital regions that become incorporated. And this is called reserve capacity. So as a task gets more complex, more brain regions get incorporated to keep performance at a higher level. If you look on the right, people with HIV infection and cognitive impairment at baseline, they don't have the same brain regions activated. If you look at tracking 3 balls, that practice effect doesn't occur. In fact, you have diffuse activation all over the place. And then finally, if you look at the most difficult task, you can see activations diffusely throughout the parietal, temporal, occipital cortex, but frontal regions really aren't getting pulled in to the same extent as a normal individual. So this is an example where even more reserve capacity is being pulled in, and these individuals make a lot of errors. So they make a lot more errors, they're using a lot more real estate to try and get the job done. That results in a very high glycolytic rate. So it makes some sense that glycolysis or a very high glycolytic rate is associated with an increased reserve capacity used to try and accomplish tasks that normally take less energy. Now, we did the opposite then. We looked at prognostic markers for neurocognitive improvement. As I mentioned earlier, we think it's equally important to look at what is associated with normal or improving cognition. This is the same type of analysis that I showed you earlier, where we have prognostic indicators on the right. You have the same decision tree. It produced a specificity of 96%, sensitivity 87%, accuracy 92%. We then did the same thing where we looked at longitudinal change in clinical markers, longitudinal change in biomarkers, and longitudinal change in cognitive impairment. And it actually decreased our specificity, but our sensitivity went up to 100%. So we could, relatively accurately identify people who were going to improve. And again, when we looked at the energy metabolites here, what was quite interesting was that in both models, decreased glucose and increased lactate and creatine were noted. Now, if you think about that in terms of energy metabolism, what that relates to is a push away from glycolysis into anaerobic glycolysis. Aerobic glycolysis to anaerobic glycolysis, and you're using alternative fuels like pyruvate and lactate, and this is known to be protective in a number of different model systems. So we started thinking about how could we push the brain to promote anaerobic glycolysis. And there were some very nice talks yesterday with exercise from Toronto and Colorado, amongst others, that I think very nicely showed high-intensity exercise is beneficial. If you exercise to exhaustion, you can produce lactic acid, and that will, from your muscles, and that very easily crosses the blood-brain barrier. But in all reality, we're not going to do that, right? People are not going to exercise to exhaustion on a daily basis. I know I won't. So we thought that the intranasal delivery of insulin could be a way in which we promote the utilization of available glucose, and then the brain has no choice but to turn to alternative sources of energy. This is something that's been done quite extensively over the years. I think the earliest was 2004. Intranasal insulin has been delivered to a whole variety of patients in a whole variety of doses that really don't seem to have any rationale. But in every case, the safety and tolerability is very good. And even in normal individuals, intranasal insulin improves cognition to some extent. The way this works, you can see on the lower left there, you use a delivery device that I'll show you a picture of in a second. It delivers a particular particle size and a mist to the cribriform plate. From the cribriform plate, it goes up into the olfactory system and then rostral into the brain parenchyma. And it has very good distribution throughout the brain, and importantly, it doesn't get into peripheral circulation. Of course, if you're not diabetic and you get an insulin bolus in your periphery, you're going to have a glucose crash. So this is one way of selectively delivering insulin into the brain. Most notably, there was an Alzheimer's study that was very effective, conducted by Suzanne Kraft in a small number of subjects that has now ramped up into a multi-center trial. Hopkins is one of those performance sites. It's a little early to conclude anything from that study yet, but at least in the early study, it was very beneficial. So we have an intranasal insulin trial as part of our center that's ongoing. We just, as these things go, we began this 3 years ago or so, and between funding and everything else, it took a while to get going. We've now enrolled 10 subjects, 2 of them completed. You can see the device on the lower left. There's 2 different flavors of these, at least major manufacturers. One is Impel. This one is a mechanical device. You just simply insert insulin into a cartridge, hold it up to your nose, and the way that the ergonomics of the nose piece is, it hits the cribriform plate. You simply press the button. And then there's another one sold by Curve Technologies, which is electronic. Both work equally well. What I want to leave you with is something I think that's quite interesting. So as part of this study, we began to think about, what is insulin doing to energy metabolism in the brain? And as I mentioned earlier, there's really not much known about insulin and energy metabolism in the brain, right? Insulin's anti-inflammatory and neurotrophic. So we did some intranasal delivery into a mouse, into lots of mice. And then initially what we did is some RNA-seq, basically a whole genome sequencing. And we came up with, we looked at about 21,000 some odd genes. Following intranasal insulin, you see changes at 15 minutes, 1 hour, 6 hours. About 200 to 300 transcripts are changed. Some of them are elevated and stay elevated. Some go up and down. There's some interesting patterns there. But what's really interesting to us that came out of this was that glycolysis wasn't really on the map. We did see, and I'm not going to show you the data, we did see a decrease in glucose, a very rapid decrease in glucose. But we saw genes upregulated that were related to beta oxidation of fatty acids. And we thought this was very interesting because the brain's not supposed to do that, right? The brain uses glucose. That's the primary fuel that the brain uses because it's readily available, it's rapidly made, and as far as we know, beta oxidation doesn't really occur. We then started culturing neurons and astrocytes to try and figure out which cell type was doing this. These are a number of genes that are related to, that little square is beta, to beta oxidation. You can see there's a lot of genes related to beta oxidation that go up in astrocytes, surrounded by red there, anywhere from 1-fold to 6-fold, depending on the time frame that you look at. But nothing happens in neurons. So neurons see insulin, we see a lot of gene changes, but none of them are related to the beta oxidation of fatty acids. We validated a bunch of these by secondary means. And what's really, I think, important about this is that glycolysis can produce 4 molecules of ATP per cycle, and it uses 2. So there's a total production of 2 molecules of ATP. Beta oxidation, which mostly occurs in the peroxisomes and mitochondria, can produce, in this case, palmitate is the example, 131 molecules of ATP and uses 2. So it's about a 50-fold increase in energy production. Now the caveat is that it's slower, much slower than glycolysis, but obviously the energy yield is huge compared to glycolysis. We looked at fatty acid utilization, which is how this occurs. in astrocytes, and I'm just sort of going to give you the gestalt of this. At top, the number 18 is the carbon chain length, and then the number after the colon is the number of saturated bonds. So you can see that there is a desaturation, an elongation to 20, a desaturation, an elongation, and a desaturation, which is very indicative of usage of fatty acids for energy metabolism. And if you look at the bottom, that's an example of a seahorse study, which looks at various measures of mitochondrial function. The big peak there corresponds to total respiratory activity. So if you add insulin to astrocytes in the presence of some fatty acids, you see a huge increase in total respiratory capacity. So to summarize, we see that, and we heard this a lot yesterday, metabolic abnormalities are very common with HIV infection. They're associated with cognitive impairment as well as other peripheral outcomes that themselves could be related to cognitive impairment. The contribution of HIV versus antiretroviral therapies I don't think is that well delineated at this point, but suffice it to say there's some contribution of both those. Often these changes can be subclinical, that is, they don't reach a threshold to be called insulin resistant, but nevertheless, those subthreshold changes are associated with cognitive declines over longer periods of time. If you are able to promote anaerobic metabolism, that looks like it's neuroprotective, and we think that the intranasal delivery of insulin could be a protective strategy in this scenario, and surprisingly it shifted the utilization of energy in the brain to a very prominent use of beta oxidation of fatty acids, and of course that produces a lot of energy. Let me just, we have a fantastic team at Hopkins, a lot of great collaborators that are very generous with time and samples, including UCSD, Mount Sinai, the MAX cohort, the Weiss cohort, and thanks to the NIH for funding, especially to fund our Novel Therapeutics Center, and last but certainly not least, the fellows in the lab who do all the work. So, thank you.